Okay, it says the bases in DNA nucleotides contain nitrogen. Researchers grew bacteria on a medium containing 15 nitrogen or heavy nitrogen. It's put there in brackets. So it's been kind of helpful, the question. For several generations. They then transferred the bacteria into a medium containing 14 nitrogen. This is ordinary nitrogen, or we call it light nitrogen. They analyzed DNA from the bacteria at three stages. Whilst the bacteria were growing on the 15N medium, after one division of bacteria on the 14N medium, and then after two divisions of bacteria on the 14N medium. The diagram shows the results. Okay, so we've got this diagram. It says bacteria grown on 15N medium. Bacteria are then transferred to a 14N medium, and this is what the DNA looks like after the 14N medium, and then DNA after two divisions in the 14N medium. So only one division. Well, the original bacteria is grown in the 15N medium. It's then transferred for two divisions into a 14N medium, which is the light medium. Okay, it says describe how the proportion of DNA that contained 15 nitrogen changed at each division when bacteria were grown on the 14N medium. Okay, so over here we had 100% of bacteria with 15N. Okay, both of these strands are 15N. Over here, we only have 50% of the DNA that has the 15N, um, that has the 15N DNA from the medium. Okay, because the lighter ones are the 50 N DNA, and we only have 50% because there's only four strands here, and two of them are 15 N ones. Over here, we have only one, and we have two. Okay, we only have one and two. We have two out of 1, 2, 3, 4, 5, 6, 7, 8. Okay, we have two out of, there's 8 strands altogether. These are 4 double helixes. Each double helix has 2 strands of DNA. So there are 8 strands of DNA, and only 2 of them have 15 nitrogen isotopes. Okay, so 2 out of 8 is a quarter, or 25%. It says describe how the proportion of DNA has changed through the divisions. Okay, so over here it's gone from 100% to 50%, and then it's gone from 50% to 25%. Okay, so as we can see, the proportion of DNA decreases by 50%. Okay, it always decreases by 50% after every division. Okay, so we would say that it decreases by 50% after each generation. Okay, that's what we would say for this question. Okay, looking at the mock scheme, it says decreases by 50%, which we got per generation or per division. Okay, which we said after each generation. Okay, and that would be worth 2 marks. Okay, moving on. Next part to this question, it says the change in the proportion of DNA containing 15 nitrogen is due to the way in which DNA replicates. Explain how. So as you can see, we saw that we had 100% of DNA with 15 nitrogen. Over here, it became 50% of DNA with 15 nitrogen, and over here it became 25%. Okay, so each time it's halved by 50%. So this links to the way that DNA replicates. Okay, why does it halve each time? Okay, if we're growing in 49, well, it's because DNA replication is semi-conservative. Each strand of DNA acts as a template, and then the new strands of DNA are synthesized, okay, in that particular medium. So in this medium, we have this strand of DNA that is synthesized containing 15N and 15N. Okay, these two split. Each one acts as a template when they are replicating again. But this time they don't have any more 15N. They're in a 14N medium. Because they're in a 14N medium, you will have one original strand of DNA, which was 15N, and then you would have one newly synthesized strand of DNA, which is 14N. Okay, So that's basically how we answer this question. Okay, so we would say that DNA replication is semi-conservative. So DNA replication is semi-conservative, and because it's semi-conservative, we know that it's semi-conservative because each DNA strand acts as a template. Um, so after the first, each strand is synthesized in the 14N medium. So after the first, each strand is synthesized using 14N medium. Okay, and if we look at the moxium, it says in DNA replication, strands separate. Okay, each acts as a template, which we had. One strand in each new molecule, semi-conservative replication of DNA, new strands made using 14N. So we have 3 marking points that we only needed 2. Okay. So it says next question now. Okay, we've got 3 more questions. Okay, it says explain why the replication of DNA is described as semi-conservative. Okay, and this is just the terminology that you need to get used to. We've used the template strand quite often, and that's what we're basically going to say. Okay, so DNA is described as a DNA replication is described as semi-conservative because each strand acts as a template. Each strand acts as a template from the original sort of parent strand. Okay, so each strand acts as a template. Because each strand acts as a template, one strand will be from the parent strand and one strand will be newly synthesized. So one strand Okay, that's how I would answer this question. Okay, looking at the mock scheme, each strand is copied or acts as a template. Always mention template because that's normally always a mark, okay? And then DNA, one new strand and one original strand, which we said, and we said parent strand. Okay, so that would be worth 2 marks. Okay, next question. It says over here, bacteria require a source of nitrogen to make the bases needed for DNA replication. In an investigation of DNA replication, some bacteria were grown from many cell divisions in a medium containing 14 nitrogen, a light form of nitrogen. Others were grown in a medium containing 15 nitrogen, a heavy form of nitrogen. Some of the bacteria grown in a 15N medium were then transferred to a 14N medium and left to divide once. DNA was isolated from the bacteria and centrifuge. The DNA samples formed bands at different levels as shown in the diagram. Okay, so DNA from the bacteria grown in the 14N medium, the DNA is right at the top. DNA from bacteria grown in a 15N medium, the DNA is right at the bottom. And then DNA from bacteria grown originally in the 15N medium, but then transferred for one cell division in a medium containing 14 nitrogen, it's in the middle. Okay, and that's because if I explain it before going on to the question, if you grow bacteria in a 14N medium, the 2 strands of a helix will have one will have 14N and the other will have 14N. So this is really light. Okay, this is really light. That's why it's going to be less dense. Because it's less density, it will just float at the top. This is very heavy because this is going to give 15N and 15N. Okay, so this is heavy. Okay, that's why it sinks to the bottom. It's right at the bottom. And then finally, this final one, it's going to be a mixture. It's going to be 14N and it's going to be 15N. So this would be in the middle. Okay, not heavy or light. Okay, the density will be in the middle. That's why it sits there in the middle. Okay, so what do tubes A and B show about the density of the DNA formed using the two different forms of nitrogen? How would you answer this question? Well, we can say that it says that you have to compare what do they tell you about the density of DNA formed using the two different forms of nitrogen? We can say that 15N has a greater density because the 15N causes it to sink to the bottom compared to the 14N, which allows it to float at the top. Okay, um, so we can say that 15N isotope has a greater density. Okay, there we go. Explain the position of the band in tube C. We can say that DNA we've already mentioned it, that one strand will have 14N and one strand will have 15N. So all we need to say is that DNA, well, one will have the 14N strand, one will have the 15N strand. And basically that's a mixture of the densities. That's what we're going to point towards. Okay, so we would say that one strand is heavy, contains the 15N isotope, and one strand, and this is going to be the newly synthesized strand, contains the 14N isotope, and therefore the densities are balanced. Okay, looking at the mock scheme, it says DNA with one heavy and one light strand, which we've also said. Um, it says new, which we said newly. We didn't say one light strand, so you might want to mention one light strand. We said synthesized strand. Okay, made with 14, and we've also said that. Okay. So there are keywords. Moxie was a bit picky, to be honest, for 2 marks. I would only give that question 1 mark, but it was really picky with the mark scheme. Okay, and then finally, final part of this question. It says, in a further investigation, the DNA of the bacterium was isolated and separated into single strands. The percentage of each nitrogenous base in each strand was formed. The table shows some of the results. We need to fill in the table using our knowledge of base pairing. So we know that A pairs with T, and we know that C pairs with G. Okay, so if this is strand 1, okay, first of all, there's one missing number over here, and we know that the percentage is always out of 100. So if we add into 26 at 28 at 14, that's gonna give us an answer of actually I'm just gonna use a calculator. So we have 26 at 28 at 14, that's gonna give us an answer of 68. So the leftover percentage is going to be 32. Okay, so cytosine is going to be 32. Now this time, that's strand 1. So strand 2, the cytosine pairs with the guanine. Okay, so the amount of cytosine present in strand 1 should be the same as the amount of guanine present in strand 2. This should be 32 as well. because 32 guanines will pair with 32 cytosines, because cytosine is present in one strand, that's 32%. So that means in the opposite strand, there must be 32% of guanines. So 32% can pair with 32%. Okay. If we have a look, we know thymine, we have 14% in strand 1, and we have 14% in strand 2, because they pair together. We have 14% of adenine in strand 2. Okay, we have 28% of guanine in strand 1. Guanine pairs with cytosine, so that means in strand 2 we must have 28% of cytosine. Okay, because we had 28% of guanine in strand 1, that means the 28% needs to pair with something, and G can only pair with C, so in strand 2 we must have 28% of cytosine. Okay, and then finally, the thymine, over here, the adenine has 26% in strand 1. Because it has 26% in strand 1, it can only pair with thymine. Okay, so there must be 26% of thymine present in strand 2. Okay, so this was actually a really confusing table, but it's just about using your logic about what you already know and applying it to the question. Okay, just remember strands 1 or 2, 1 and 2 are opposite of each other, like we have over here, and they're going to pair opposite each other, just like that. Yeah, so whatever you have in strand 1, the opposite letter must be this of the same percentage. Okay, and there we have 32, 28, 32, 26.